Ah, yes, the cell cycle. In this video, I am continuing my molecular biochemistry series, and we are detailing the cell cycle. In previous videos, I talked about the molecular structure of DNA. I touched on how we can synthesize the nucleotides of DNA, and then we even talked about DNA replication, which is the necessary precursor for a viable cell after undergoing cell division. And this DNA replication process takes place in the S phase of the cell cycle. But from a conceptual standpoint, we will begin with the G1 phase. And just a quick overview, I'll highlight each phase. So the G1 phase is a phase where the cell's cytoplasm gains volume, the organelles become larger, and once a CDK signals that it is time for the S phase, DNA replication takes place, and then the S phase heads to G2, where the cell again grows larger in size. And finally, we enter the M phase, which is the process of cell division, such as mitosis or meiosis, and the cell physically divides into an additional cell. So that's a high overview. We now will look a bit closer at the G1 phase, which I noted is cellular growth. There are different types of cells in our body, and some of our cells are called labile cells. These cells are continuously undergoing the cell cycle. They are growing and dividing over and over and over again. They are constantly doing this, and an example is our skin cells are constantly undergoing this cycle. And we also have something called permanent cells. Permanent cells exit the G1 phase into the G0 phase, and we call this cell arrest because these cells will not divide. They will not undergo the cell cycle. They will not move through the interphase, which is just G1, S, and G2, and they will not divide. For example, neurons, once we're done with our developmental period in our lives, we have a set number of neurons. We do not make more of them. They are surrounded by something called glial cells that act like cushions and do various different things around neurons, but the neurons themselves are fixed. That is why neurodegenerative diseases are inevitable in our lives, and it's a fascinating subject area to study and think about techniques where we can maybe prevent this or reverse any neurodegeneration that takes place. Additionally, we have red blood cells. These do not divide either, but they are produced in the bone marrow throughout our lives. We also have cells called quiescent cells. These cells arrest into the G0 phase just like neurons or permanent cells, but they actually are able to re-enter the cell cycle and undergo replication and division. For example, liver cells such as hepatocytes or immune cells like lymphocytes undergo this quiescence. So once the CDK signals as like a checkpoint that we can enter the replication phase, our cells will begin replicating. And I don't need to detail DNA replication, but there are a couple of regulators or factors that kind of regulate this process. Now, there are two options. We could either overactivate CDK. These are called proto-oncogenes. They activate CDK, which activates the cell cycle. And this can cause cancer because we may cause the cell to be dividing too often and too frequently without stopping, and this can lead to tumors. Alternatively, we have these genes called P53, which activates P21, which inhibits CDK and prevents this cancer from happening, and these are called tumor suppressors. But DNA damage or something like HPV can inhibit P53, and so P21 is not able to inhibit CDK, and that results in the cell cycle proliferating once more, and this is cancerous. So those are our two options for thinking about cancer. Once we finish DNA replication, our cells, I keep saying we, like we are a cell. I guess we are technically a cell. But anyways, we enter the G2 phase, and the cell will once again grow and get larger before it is undergoing cell division, and this is meiosis and mitosis. We have the prophase, the metaphase, anaphase, telophase, and then finally cytokinesis, where the cell physically divides. This is the first lab technique we'll talk about in this series, called karyotyping. And what it is we can add something called colchicine, and we can add this to a culture of cells, and this will halt metaphase. And in metaphase, we can stain the chromosomes, and then we can analyze them for any abnormalities. And this is useful for identifying any disorders or diseases or various different genetic abnormalities. That's a great overview of the cell cycle. We need our cells to replicate in order to sustain our tissues and our organ systems. And it would be helpful if our neurons could replicate, but they do not, and inevitably we can get neurodegeneration. Alternatively, we have the cell cycle checkpoints, which if they become overactive or are unable to be suppressed, we can have cancer. So I'll see you guys in the next video. Take care.